All right, everyone. So welcome back. We are in chapter 12 and 13 today. Let me get position here because it's a little tight in this area. So let me kind of reorient myself so that we can start talking. Um, I'm hoping that these videos will actually be available to you guys before your second exam. At least at the time of this recording. For you guys in the future, you hear me say this all the time. I'm a broken record, but these should be available to you guys already. All right, so let's get started. What did we talk about the last several videos? And let me just kind of get ready. I should probably do this before recording, should I? Anyways, what do we want to talk about? Today, we are going to be talking about the cell's life cycle and, to an extent, the process of asexual reproduction called mitosis. Now, I'm going to use the term asexual and sexual reproduction because asexual reproduction is the first one that we're going to go through, which is mitosis. And then sexual reproduction is the second one that we're going to go through, which is called meiosis. Now, unfortunately, I usually do this lecture on a whiteboard just because I find it's very simple to grasp concept as you are drawing it because you have that tactile memory. You know, you're using your muscle memory to kind of aid in the understanding of this concept. So for those of you guys who are viewing this video, um, come to lecture. That's the best thing I can say. Come to lecture if you can and watch how I do it on the board. I can't do it on here all too well just because I have a mouse and I try to do a lot of drawings, but it turns out really, really bad. I might be able to post up an older video, but there are certain things that I said in that old video that is slightly too basic and you guys would need to know more. So I'm kind of hesitant to show the intro videos to you guys just because of that. So if anything, come to lecture. If we've already had lecture, then ask someone for their notes if you weren't able to attend. But let's get on with it. So we talked about the cell's life cycle, or we talked about the cellular respiration. We talked about photosynthesis. We talked about how proteins are involved. And now, in the last chapter, we talked about cell signaling or the communication of the cell. And so here's a question that you guys, I'm hoping as we're going through these chapters that you're kind of just thinking about. You don't necessarily have to ponder on it, but just have a thought, right? Just have like, hey, how do cells communicate to each other? And why do they communicate and when do they communicate? And if you guys can start understanding that and know that all the metabolic functions within the cell requires ATP, so cellular respiration is still being used everywhere, we're making those ATP for a reason. Now, when a cell is utilizing energy, it's got to be efficient about it. It's got to be organized about it. And so, if another process is taking precedence, then hopefully the communication between the different organs of the cell, the organelles of the cell, is able to function properly. And to recall back in chapter 8, remember proteins are what moves the cell. Protein concentration. Yes, the DNA codes for the protein, but ultimately it's the protein at the very end of the central dogma that builds that characteristic of the cell, that builds the ability to replicate new DNA, replicate more RNA, replicate more proteins. It is, we need those tools in our cells so that it works properly. And so, in this understanding, if we take a look at this slide right here, and you guys can see, we're undergoing cell division, cellular division. And that requires a lot of synchronization between the nucleus, the mitochondria, the apparatuses, the ER. It requires all of that communication efficiency so that it can divide properly. And if it doesn't divide properly, that's when we come to issues where cell malfunctions, toxicity builds up in the cell, or we start developing cancer. And cancer, we'll touch on in this chapter, but it's not too um, prevalent in this chapter. We do touch on it, but we do have a whole chapter that we're going to be talking about towards the end, I think, either in this class or in your future classes. And so we're going to go from there. Okay. So the idea is we're starting from two cells and going into four cells. So there are a couple things. Because we're talking about cell division, we also have to kind of include the idea of the cell's life cycle. Now, just like with humans, just like with any living creature, we have phases in our development. And the cell is going to go through their own phase of development too. So during the time that they're ready to begin the replicative process or begin the growing and dividing, um, it will go through an intermediary phase to prep for that stage. So, the analogy that I like to use, right, is say that you have a home right now, or say that you have an apartment or a dorm room, right? And someone gave you another exact size place. So that means you have two locations. And your goal for this is to be able to make this second place exactly identical to your first place. Because again, you don't want things to change. You're a creature of habit, so to speak. So you want everything to be exactly as how you imagined it in the first apartment and then identical to that in the second apartment. So the question then comes, what do you do? How do you get the second apartment to be identical to your first apartment or second dorm room to be identical to your first dorm room, right? What do you do? Well, first things first is you got to purchase the same furniture, right? That it's a blank apartment. You got to get that to be exactly the same. So you're going to go out, you're going to purchase some brand new furniture. Great. Now, the thing is the furniture already assembled for you, or is it going to be shipped to you in a box and then you have to carry that box over to your next apartment and then unpack everything? And most of the time, especially if you go to IKEA, for those who don't know IKEA is I think it's a Swedish furniture company right here down in the South OC area. But anyways, if you purchase these furniture, you're going to get it in a box. And then if you have it in a box, you're going to take it to your apartment. You're going to carry it in a nice organized package to the new place. And then from there, once you have it, then you start unpacking. Then you start reshaping the room to exactly how it was in your area. And then from that point, now you have two identical places. So here's the thing. Okay, again, this is analogy still. Here's the thing. What happened? Well, you had to double everything, right? You had to grow and double everything. You had your living space grew in size, right? Now instead of 100 square foot, you have 200 square foot because it's the exact same dimensions. Okay. Instead of one bed, you have two beds. Instead of, one refrigerator, you have two refrigerators, so on and so forth. So you grew in size in terms of square footage. You doubled all essential furnitures or all the furnitures that you have in your place. You purchase the same clothes, you purchase the spoons and everything, and now you have nearly everything the way you want it. But there might be a couple things that you forgot. Oh, wait, I forgot to buy forks. Or, oh, wait, I forgot to buy extra light bulbs because I have a couple light bulbs that burnt out. Or, oh, wait, I forgot to, get dishes. So after you've done all that duplicating, all that, right? You're gonna go through that checklist one more time to make sure everything matches. And then once everything's set, then you're gonna be like, okay, we're good. All right. So that is a very, very rough analogy. And again, I'm hoping that these real world examples are helping you connect what's going to come in this chapter. Two chapters, actually. These two chapters are combined. So as long as you have that idea, the idea of we need to grow, we need to double everything, we need to make sure we have everything ready, and then we can split into two equal places, then you have a rough understanding of both mitosis and meiosis. Meiosis is going to be slightly different, but you have both understandings already. Okay, so that is the overarching concept. Now let's talk about it. So during the process of us growing and doubling everything, this is the area where it's the intermediary phase. So this is what we call interphase, where the cell is just prepping itself to grow in size, double in size, grow all the organelles, double, duplicate everything, and go through its checklist. Once all that is ready and it gives the thumbs up to continue on, then it's going to undergo cell division. Now, cell division for us, we call this the M phase. Now, M phase stands for mitosis or meiosis for chapter 13. And all it is a cell splitting everything in half into two brand new cells, in essence. So if we take a look right here, and let me pull out my little pointer here. If we take a look right here, we have the parent cell. So that's the singular cell line that we're going to look at. And then just know we've doubled everything, right? So we have one chromosome. Now we have two chromosomes. And then we split it into two equal daughter cells. That's cell division in a nutshell. Okay. Now I'm using the term chromosomes even though we haven't talked about it yet. And all chromosomes are, and again, you guys can watch animation. All chromosomes are is DNA. That's it. It's your DNA molecule. Okay. So cell division needs to not only duplicate all the organelles, but it has to also duplicate the genetic code. And that's probably the most important thing about mitosis and meiosis that we are going to be focusing on. Because once we go into chapter 14 and we start talking about the what do you call it? Genetics, we're going to be looking at chromosomes again. Okay. So in the DNA, we already know from past chapter it's a super long strand of nucleotides, right? It's a double layer. It's a anti-parallel two strands of DNA molecules bounded. Okay, and it's in a helical shape. Now, all the DNA in the cell is what we call the genome, the genetic material. Okay, and the genome consists of DNA molecules, either in a single strand or double strand, depending on the organism that you're looking at, and they're going to be packed up into chromosome. Now, analogy time. So again, analogy time. Let's shift over. If you can imagine DNA like your Christmas lights, okay, so just take a moment, just think about it. How, after the holidays are done, okay, what do you do with those lights? And I know this is a very, very weird example, but bear with me. What do you do with those lights? Well, for the most part, we take them down, right? Now, if you're really crazy, like myself, we keep them up for the whole year, and then we would be like, hey, we can ready to put the lights up. But for most of normal people, I still have my lights up. But for most of the normal people, um, you would take them down. Now, how are you going to repack your Christmas lights? Well, you either take them all down, shove it into a big, massive spaghetti ball, and throw them in a box and pray next year that they're not tangled and you have a good time opening it. Or B, you wrap it around some sort of plastic, structure where you can wrap it around, or you wrap it around your arm just to form those little loops. And then you pack them in a nice, neat pile. And then you put them in a box. And your DNA is like those Christmas lights. They don't want to be thrown into a box all spaghettified. And then when we need to look at a specific sequence on the DNA that's all tangled up and it's sterically hindered or it is strained and then it might snap, we don't want that. We want something that's easily accessible or able to organize it, right? And then read it when we need without causing any other issues on the DNA molecule. So your DNA, your chromosome, the reason why we call them chromosomes is because your DNA molecule has a structure to it. The double helical structure is the pure DNA, but what the DNA wraps around is what we call a protein histone. Now, when the histone has the DNA wrapped around it, we call this singular unit a chromatin. Okay, so chromatin. And then we pack all those units, zigzag them together, and then that condenses into a chromosome. So here is an example of what a chromosome will look like in the DNA molecules. Okay, so chromatin again is just the DNA with the histone protein and it's packaged fully. Okay, now for all cells that are utilized in the body, we would call these somatic cells, with the exception of our reproductive cells, which is the sperm or the egg. Now, we are looking at the sperm or egg cells. We don't call these somatic cells. Okay, somatic cells is every other cell in your body. The sperm and the egg cell is what we call gametes. Okay, and the reason why we call them gametes is because in a somatic cell, you have your full set of DNA, but in the gametes, you're going to have half the amount. Now, why is that? Okay. And the reason why we have half the amount is because we know through health class, through past courses, that in order to create a living embryo, right, you need two things. You need the egg and then you need the sperm, and they need to come together and fertilize each other. Well, if we had a full set of DNA in the egg and a full set of DNA in the sperm, and then we come together, then we have double the amount. We don't want that in our children, right? We don't want to have too much information, right? Otherwise, your body's going to be undergoing analysis paralysis kind of thing. It's just not going to work because there's too many instructions going on and you just freeze, right? So, our body, at least in terms of human biology, did a wonderful thing. If we develop the sperm, we're going to take only half of that DNA. If we develop the egg, we're going to take only half of that DNA. And then when two halves come together, then it makes one whole. And so you have one complete set of DNA, one from mom, one from dad, complete set of DNA, and you don't overload the system. Okay. So now we're going to talk about the shape of the chromosome. Initially, before any duplication has occurred, we generally will have an S-shaped chromosome. Now, I like to use letters just because it makes things easier for students to understand from my experience. So your chromosome is going to be a single-stranded chromosome, and if I can draw it right here, it's going to look something like this. Okay? It's just a singular strand. It's condensed into a single strand, and you're good. Right? Now, that's one chromosome. Okay? When it duplicates, we will need an exact identical copy of the S chromosome or the single-stranded chromosome. So if we want to build the exact copy, what we're going to do is we are going to duplicate that using DNA replication, and we are going to make sure that they're still stuck together. Because again, we don't want other pieces to fly around. We don't want the duplicated strand to fly around because your cell's not going to know, hey, did we duplicate this yet or not? So we're going to keep them condensed and we're going to keep them stuck together, right? In a singular unit. Now, this is what we call our X-shaped chromosomes. So S shape is single stranded. X, the letter X shape is the duplicated strand. Okay. And at the center, we call this a centromere. Now, mere representing unit of, like polymer, right? Polymer, monomer, MER is unit of, centro, center, right? So this is the unit at the center. In essence, that's what centromere stands for. It's a unit at the center that holds the duplicated chromosomes together. Now, we don't want to keep saying duplicated chromosomes because they're identical. So in this X shape right here, this is where we can label each strand as sister chromatids. Okay. And sister chromatids are held together by a protein called cohesin. We're very creative with the name. Cohesive. In order to keep these things glued together, we got to get a cohesive thing to to make them stuck. Okay. So the protein, which a lot of you guys are going to hopefully You're starting to recognize the pattern. Every time it ends in IN, chances are we're working with a protein. If it ends in ASE, we're working with an enzyme. If you're working with a nomenclature that ends in OSE, you're working with a sugar. So hopefully this is you're starting to see the pattern. But cohesin is the protein that's keeping the sister chromatids locked in together. Now, actually, I am teaching a mitosis and meiosis lab today, so I do have some things. So let me get it. All right. So I do have a couple models here, and we can see right here. I'm hoping you guys can see that. Okay, can you guys see that? Okay, I know it's a little small, but this is an example of a single, let me see here, single stranded chromosome. Okay. Notice the center and notice that it is S-shaped. Okay. So when we duplicate it, again, there's going to be a band right here of protein called cohesin, and we are just going to lock them in together. And now we have a duplicated sister chromatid, right? So each band that we see are sister chromatids here. Okay. So that is an example of a duplicated chromosome, X-shaped chromosomes. I know it's kind of hard to see. I kind of shrank my face down really small. Hopefully it helps. Okay. So there we go. All right. So watch these videos. It helps. During mitosis, the main thing is to keep track of your chromosomes. Now, when they're in this X shape, this is a single strand of DNA. So this is one molecule of DNA because they're still combined, right? They're not two different units yet. So because if you count the DNA molecules and there's only one whole DNA molecule here, we consider this as one whole chromosome. There are two chromatids in this structure because there's two strands. Okay. Now, when it separates, now each individual piece is a chromosome, but they don't have any chromatids attached to them. So this is two chromosomes right here, zero chromatids. One chromosome, when they're attached, two chromatids because we see the X shape. When they split, this is two chromosomes, right? No chromatids because they're not in their X shape. Okay? So write that down. All right. Cytokinesis is a step after mitosis. Cytokinesis means the cell's going to move apart, they're going to divide, and then the cell's just going to start living. Okay. Meiosis is the division for the gametes. Okay. And we'll talk more about that later on. So the cell cycle consists of the interphase and the mitotic phase. And in this interface, we can divide it up into three new stages. Okay. I wish they said stage, but they're using the word phase. So just be mindful. Okay. So again, what's the idea? Go back to the analogy of our apartment, right? We got to grow and double in size first and then duplicate everything. So G1 is the first growth phase or the gap phase. Then we have the synthesis phase, S phase. Guess what we're synthesizing? What's important? Your DNA. G2 is the last growth phase, okay, or second gap phase, and then the M phase occurs. Now, these three together is what we call the intermediary phase or the interphase, okay? Now, there is a logic behind all of this, but just know that in these phases, pay attention to where the DNA is being duplicated. In G1, there is no duplicated DNA. So what should the shape look like if the DNA condenses? Should be a single strand. Once it goes through the S phase here, at the beginning, we're going from one molecule to hopefully about two molecules. Okay, at the end, it will be two strands of chromatids, right? In the middle, you're starting to make it. So you can think of it as from one to two, and as we progress through the S phase, it goes by fractions at that point. Now that's confusing, so don't worry too much about it. It's just a process. Okay. But just know at the end of S phase, we have fully duplicated DNA so that we have a sister chromatid, duplicated sister chromatid. And then once we get to G2, it's still a sister chromatid, right? Nothing, it didn't split yet until we hit mitosis where we split the chromosomes from each other, or the chromatids from each other. Okay? So here are five stages that you would need to know. Now, there are going to be key events for you guys in each of the stages. So here I'm going to explain briefly, verbally. Writing it out is actually really messy. This is my second time recording this video, and it just got way too messy. So I'm gonna do something, and I'm gonna hopefully verbalize it for you guys in video. And then when you come to lecture, I'm going to write it on the board. Okay. Now, the main stages that I usually focus on is prophase, metaphase, anaphase, and telophase. Now, in your textbook, we also go through the pro-metaphase just because it is a transition from the prophase and metaphase, and there's certain things that happen in there, so you do need to keep an eye on it. But when I'm drawing it on the board, I'm only going to go through the main four. Okay, the pro-metaphase is like that in between, but your textbook considers it as a unique stage. So acronym is PPMAT. It used to be PMAT, but now with pro-metaphase is PPMAT. So if that helps. So here we go. Let's talk about it. In prophase, we're going to take a look at it visually here, and I'm going to verbally tell you. In prophase, or let's right before mitosis begins. Okay, your DNA again is a spaghettified mess, right? Now, before we start moving, we have all of our stuff in the house. We haven't packed them up into boxes yet. But once we're ready to split and move everything, then we need to pack it up. So during the interface, everything's just everywhere, right? We're living, we're running through our normal day. And once the cell knows, hey, we're about to move, we need to split, then it starts packing. So in G2 of the interface, your DNA is not condensed. Once prophase begins, the DNA will now condense. But where is the DNA? It's in the nucleus. And you guys know from last chapters that the nucleus is a double membrane. It's hard to get things out if you're stuck behind a double membrane. Just saying. So your cell during prophase, the key events that are going to happen is the nucleus will begin to disintegrate. Now, what's holding the nucleus together? Remember that it's intermediary filaments, it's the cytoskeleton. So we're going to start degrading all of that so that the nucleus is able to get released, right? The DNA inside the nucleus, the chromosomes inside the nucleus can get released. Now, if we take a look at this image right here, how many chromosomes do we see? Well, we see one, we see two, we see three, four, five, six. There are six chromosomes here. Okay. And if we take a look, there's also these other helper called the centrosomes. Okay. Now, don't get this confused between centromere, because the centromere is the middle point of a chromosome. The centrosomes are different helper proteins called centrioles. And then from the centrosome, okay, you're going to see your cytoskeleton, aka the microtubules, start expanding from the centrosome. So that is all the key events that happens during prophase. Now, in the prometaphase, okay, what's going to happen is the nuclear envelope further disintegrates, okay, pretty much disappearing. The spindle fibers, aka the microtubules, which we call them spindle fibers in this case, begins to fully extend out to the rest of the cell, and then we can start repositioning our chromosomes. Now, why is it that the cytoskeleton microtubules need to extend out everywhere? Again, recall back to past chapters. What allows things to move? What moves the vesicle, right? What moves protein from one end to another? They don't just, some of them will free float, but most of them are not free floating. Just like us driving on the road, we're not driving randomly. There is an organized manner, and that cytoskeleton is what keeps everything in organized, right? In order. So these spindle fibers that are extending are going to also have motor proteins that move the chromosomes to a designated area. And the designated area is what we call the middle plate, the equatorial plate, the metaphase, meta for middle. Okay. So you have the metaphyseal plate where we're going to line the chromosomes up directly on it. The spindle fibers are now connected to the chromosome. Okay. At the centromere. Okay. And we are going to perform some sort of action. Okay. So just imagine, okay, if we take a look again, look at the position of the centromere. What happened? Well, during the pro-metaphase, they started shifting. The spindle fibers extended out. But look at the position where they're shifted towards. They're shifted towards the polar ends of the cell, north and south, east and west, and then all others in between. So they're directly opposite of each other. Now, why is that? Well, again, think about it. It's trying to split into two cells. So they're going to split apart. Now, the best way to split something apart is by pulling from opposite ends, not together, right? If you split on both on the same end, it's just going to be pulled in one direction. So if you want to split something equally, opposite ends. And so in metaphase, the centrosomes are on opposite ends, the chromosomes are in the middle, and it's getting ready to begin splitting. Now, during anaphase, the splitting occurs. But remember, what's holding your chromosomes together? What protein? Okay? Your cohesin. So during anaphase, there's going to be an enzyme coming in to cut up the cohesin so that we can split them apart. Now, this enzyme ends in the letter ASE, and its main function is to separate these chromosomes apart. So guess what we're going to call it? Separase. Separase is the enzyme that separates the cohesin, the chromosomes from each other by digesting the cohesin or splitting the cohesin. And then at the very end, once the chromosomes are on opposite ends, we can isolate each of those groups by reforming the nucleus. Okay, that's what telophase is all about. It's reforming those nucleus, disintegrating the spindle fibers because we don't need them anymore. Remember, back in cytoskeleton, when we talked about it, microtubules and actin filaments, the microfilaments, do not stay around for too long. Intermediary filaments are the ones that usually stay around for a while. But the microtubules and the microfilaments, they're there when we need them. Otherwise, we're going to pack them away. And so once cell division is completed, we don't need them anymore. So we're going to start removing all these microtubules, right? Reform that nucleus with intermediary filaments, and then boom, we have two distinct cells. Now, after all those phases have done, the last step is cytokinesis. Cytokinesis is technically not part of mitosis, so please be wary of that. Cytokinesis, use the term cyto meaning cell, kinesis pertaining to movement, kinetics, right? Kinesis is the splitting of the cell through movement. Now, how does that work? Well, in order to split the cell easily, we're going to pinch off the cell while it's being pulled apart. And then you're done. Okay? So watch an animation. Again, I'm not going to show the animation here just because I don't want YouTube to flag me with copyright. Because this is a publisher's animation, so they've already, yeah, I'm not going to show it. So you guys can watch it on your own time. But you should be able to find it. So again, we've talked a lot, and I did a lot of verbalization, and I'm hoping this makes sense. So these next several slides, we're going to speed through it. Just FYI right now. So mitotic spindle fibers. They're fibers that are used in mitosis or meiosis. Okay. Centrosomes. Again, these are where the spindle fibers are extending from. But what I didn't really call the structure of what is being built from is what we call an aster. Now, an aster, best way to think about it is just an array of spindle fibers. That's it, an array. And an array is just like how sun has rays emitting from it. So spindle fibers are emitting from the centrosomes. Okay, so we call these asters. Now, the question is, where do the spindle fibers hook onto? Well, the best way to split these chromosomes is not to pull it at the edges. Because what happens if we pull at the edge, we break them apart. That's not good. We want to pull them at an area where they won't break as easily. And because we want them to be equal size DNA molecules, best place is to do it right in the middle. So they're going to hook on, the spindle fibers are going to hook on to the centrosome because that's in the center, that's in the middle of the chromosome, and they're going to digest the cohesin using separase and then split them apart. Now you're not putting any strain on the floating DNA molecule. You're not grabbing it by the leg here and then yanking it too hard where it splits into two uneven pieces. Albeit sometimes it does happen, and this is where errors occur. Okay, and sometimes this is what causes genetic defects. But we usually are going to hook them up in the middle of the centromere. Okay? Now the question is, what is this structure called? It's called a core that's for the movement of chromosome, right? Movement, kinetic. Core is the area. So we call this a kinetochore. That's it. We call this a kinetochore. So the kinetochore is a protein complex found at the center of a chromosome, the centromere, okay, where the spindle fibers will attach and pull the chromosomes, the chromatids apart. Okay. And then we can see it in this image right here. Separase separates the enzyme by digesting the cohesin or cleaving the cohesin apart. Okay. And then the kinetochore walks the chromosome. Again, we're using the road, right? We have motor proteins that are walking the chromosomes into opposite ends. Okay? And then in order to separate the cell fully, we need to form the cleavage furrow. The cleavage furrow is just us pinching the cell until it separates. So another example that you guys can try at home, and this is a great visualization and tactile memory, is try to grab a cup of flour, just all-purpose flour. Mix it in with water, make a dough. Now, your job, once you have that, is to split the dough into two even pieces without using any other tools, just your hands. Now, you guys, as you're doing this, you might try to break it apart, but then it gets all messy. But after a while, you're going to find, hey, wait, I can split it up by just pinching the dough. And if you pinch the dough and it splits, it's going to split into two nice equal balls of dough. Okay. So this is something that I encourage you guys to try if you have the time and the resources to get all-purpose flour. And if you want to make bread, it's a great way to kind of start learning how to make bread at home. So try it out, and you'll get the idea of why cleavage forms. Why is it the cell wants to do it this way? It really, really works. So now the last thing is for a plant cell, because a plant cell has a cell wall, aka an armor, um, when the plant cell undergoes division, it's going to do all the same steps with an additional step at the end. Because within the armor is expanding, but for plant cells, they need their own individual armor. So after the cleavage furrow happens and the cell splits, that in between that gap, another wall forms, another double layer wall actually forms, so that now both plants have their own individual armor set. Okay? So this is what a cell plate does for the plant cell. Okay. All right, this is a great moment to kind of pause what you're doing, just stop and kind of come back to it. Okay. I highly encourage rewatching this first half, understand some of the analogies. If the analogies are too confusing, watch the animation and then start working with mitosis and meiosis. So I would recommend at this point, draw it out, draw all the key steps. Okay. I will do this in lecture for you guys, and maybe I'll post up my intro work because I was able to utilize the pens when they provided for me. Um, but draw it out. This is these two chapters. I can't stress this enough. My old professor back in college when I took it, I think 20 years ago, told us to do it. And I didn't believe him. I really did not. And Um, told us to draw it. Our TAs told us to draw it. And ever since then, I've never forgot it. So I'm telling you guys, you don't have to if you don't want to, but if you want to speed through the studying, draw it out. It helps. Okay. And make sure it follows exactly how the video shows up. So, anyways, now that you're back, hopefully that was a good break for you guys. We're going to talk about bacteria. Okay. Now, their mode of cell division is not the same as eukaryotic cells because bacterial cells and archaic cells are much simpler. So they're going to undergo a process called binary fission. Binary meaning two, right? There's two positions, right? Fission just means it splits from a single source. Okay. That's a fission reaction. And what they're going to do is they're going to do something similar, but they're going to do DNA replication right off the bat. They don't necessarily need a chromosome because their DNA is a circular DNA. So what they're going to do is they're just going to duplicate the two DNAs at the spot while it's starting to divide. Okay. It's a much simpler process. You're going to learn about origin of replication when we talk about DNA replication. So I'm not going to go too heavy into it, but just know that DNA needs a starting site to replicate, aka an origin site to replicate, aka an origin of replication site. So yeah, we're so creative with the names. All right. So that is mitosis in a nutshell. Now, remember what I said that we have a checklist and that cells has specific what do you call it? Specific stages of their life? Yeah. So how does a cell know to go from one stage to another? How do we know that we went from infant stage to preschool stage or preschool stage to adolescence or adolescence to young adult? Like, how do we know we've transitioned from one stage of our life to another stage? Well, there's going to be certain cues, certain clues. For us, is mainly based on our developmental processes, right? So in a cell, you can think of it as something very similar. But remember, what controls the cell? Proteins. Remember, the expression of proteins, how much proteins we have in a cell will determine the characteristic of that cell in the long run. So in these cycle systems, these checkpoint systems, we are going to focus on a couple proteins that builds up in concentration and disintegrates in concentration or builds down in concentration. And based on the concentration of these specific proteins will determine what phase of the life cycle they're in. Okay. So where's the location of the checkpoints first? Well, the first one is definitely at the first growth duplicative phase at the G1. Okay, we need to pay attention to that because we want to make sure we bought everything, right? That's the first checklist, right? After that, we synthesize DNA and then we end up in the G2 checkpoint, right? Where we're finally reconfirming, double checking that we got everything ready to go before we begin moving. Okay. So G1 is that first checklist. Did I purchase all of my furniture? All right, I think I did. While you're kind of marinating on that, the next checkpoint is right before you begin moving. Am I sure that I have everything? And you go through it one more time. During the process of moving, or aka during the process of dividing, you have your M checkpoint, which is going to be like, am I moving these things correctly? Did I go to the right apartment number? Do did I do things correctly? If not, I got to go back and do it again or give up, right? And so the last checkpoint is the M checkpoint. Now, these checkpoints are so vital to a cell's life cycle that if in any way we disregard the checkpoint, then the cell is going to go through this whole cycle uncontrollably, right? These checkpoints help stops the cell from continuing on until it's ready to move on. You don't want to just be like, hey, I got to move to my apartment now. Okay, let's move without getting anything. So these checkpoints are vital. And if the cell bypasses all these checkpoints, this is where cancer comes in. That's cancer, right? Cancer is uncontrollable cell division. This is why tumors exist, benign or malignant. Cells are uncontrollably divided. And in order to divide a cell, you need a lot of energy. In order to make energy, you need carbohydrates. So there are some research done today, and they're still in the early stages of research, that some doctors will recommend, hey, if you really want to mitigate the cancer, you might want to think about the fuel that feeds the cancer, which is mainly carbohydrates, because that's what we use in cellular respiration. So that's one way to do it. Or the other way to do it is through chemotherapy, where we force a barrier on the checkpoint. And if you stop all cell division, and the cancer cell is continuously starving, right? But it can't divide anymore. It's gonna starve to death. And that's what the theory of chemotherapy is all about. So, depending on the drug, it'll either stop at G1, G2, or M checkpoint. It will tell all cells in that person's body to stop at that checkpoint. So no cells are dividing anymore. Hence why through chemotherapy, when you stop all cells from dividing, you lose the ability to grow hair, because hair is just dead cells uncontrollably dividing and it's causing it to grow longer. Okay? Nails start to kind of disintegrate. Your skin becomes a little bit more pale because, again, solar radiation damages your epidermal layer, right? You can't replenish those cells. So it's going to be more bleached, more sun bleached, because there's nothing to repair it. Okay? All right, that's enough of cancer. So, cyclin, cyclin dependent kinases, or CDKs, are the two main proteins that we're going to be looking at. Now, there's a lot of different types of cyclin. There's C cyclin, D cyclin, B cyclin, A cyclins. I just want you guys to know the main generic terms. Okay. MPFs is a cyclin that's bounded to CDK. Remember, CDK is an enzyme, cyclin's a protein, and if they're interacting together, they form a complex. Okay. And we can see throughout the stages of a cell's life cycle, based on the concentration, you can see that during the M phase, we have high concentration of cyclin and the complex, CDK complex. Okay. And then once we pass the M phase, it shoots all the way down until we start hitting the synthesis phase, where cyclin builds up again. Now, as cyclin's building up, it signals to the cell, hey, this is G2 phase. And then your cell is going to work appropriately to that, getting ready to do cell division. So that's all it is. Okay. And here's an example of what's occurring throughout. So you have cyclin degrading, CDKs are still flowing through. And then once you hit G2, then cyclin's being made again. And then we can create a mitosis promoting factor and then run through the stages. Okay. Now, last but not least, in this chapter, if for whatever reason you don't want to grow, you don't want to divide, then the cell has one last option, which is to undergo um, stasis, which is just living life without doing anything. So it's just in pure bliss. It doesn't need it to do anything, doesn't want to divide, doesn't want to grow. It's just there. And this is um, the phase where we're going to call it G naught or G0. Okay, G0 is before the G1, G2 checkpoint. It's just there to live. Okay. And so here's a summary. All right. So that is chapter 12 in a nutshell. Now, I was going to do chapter 13 here, but this is a long video so far. I might separate these videos. Just know that this is 96 slides, and I'm taking my time to explain this, so hopefully it makes more sense to you guys. So I'm gonna try to get it done within the next 15, 20 minutes. I don't know if I can, but let's get on with it. Okay. I do want this as a single video, but if it gets too long, I might split it. Anyways, chapter 13. Meiosis. So meiosis is very similar to mitosis. If anything, you already know half of meiosis, and we'll talk about that later. But just know that meiosis, because we're dealing with sexual cells, the sperm and the egg, and it's all about the genetics that gets passed on to the next generation. we need to kind of keep in mind the heredity, which is the transmission of your traits from one generation to the next. So, what are you going to inherit as an offspring? You also need to know that we want to increase variation. Again, there's a lot of genetic diseases, genetic mutations that is not favorable. So, the more variations you have in your gene, the safer you're going to be. And this whole study of both heredity and variation is what we call genetics. Okay. Now, your genes are the units of heredity. So that's, on a DNA molecule, you might have thousands and thousands of genes, and it will pretty much code for a specific trait or a specific characteristic. Okay. Now, the genes are passed on through gametes. You already heard me say it, and we have somatic cells. Now, the specific gene we're going to be looking at is determined by how far away it is on the chromosome. Now, what is a nice reference point for a chromosome? If we take a look at a chromosome right here, what is a nice reference point? The reference point, because the size of the chromosome varies, we usually look at the reference point based on where the centromere is. Remember, the centromere is a cluster of proteins, the center of the chromosome. So whether the chromosome is big or small, at least we have a relative reference point to go from. So we're always going to measure gene based on the centromere and a distance away from the centromere, which we call a locus or location. Okay? Now, in humans, we have 23 different types of characteristics or different characteristics of chromosome. We call this a karyotype. And depending on the karyotype, again, we have two sets of DNA, one from mom, one from dad. So a karyotype is essentially you're going to look at a chromosome size, chromosome shape, and you're going to match it up because mom has one copy, dad has one copy, and for us, we got both copies from mom and dad. So a karyotype is like this. So right here, I have the big red chromosome, and then I have a yellow chromosome. This is one karyotype of chromosome, right? Because one's from mom, one's from dad. It's fully duplicated at this point, right? But this is a karyotype. Okay. And we call these, because they're the same size and shape, we call these homologs. That's it. Homologous chromosomes. Now, why are we introducing this? Is because when you're looking at the karyotype, we have 22 somatic homologs, okay, or 22 non-sex chromosomes. Okay. And then we have one, which is our 23rd pair that defines the biological sex of the organism. Okay. So that sex chromosome that we look at in a biological setting is the shape, right? Why are we using the letters X and Y? The reason being is because this is an X shape. That's it. And a Y shape is literally an X shape without one leg. That's the Y shape. Okay. So for the development of an embryo, for us humans here, when we are developing, right, we generally are developing from the same blueprint, right? X chromosomes. The only difference is that because on the male uh, part of it, right, we're missing this extra leg on the chromosome. When you're missing genetic material, right, there's no proteins that can get coded. Remember, it's DNA to RNA to protein. We're missing this piece. So a certain number of proteins are not activated in the male species or the male gender. And what happens is that our cells sees that. So they're going to develop those cells in a certain way because now the protein concentration is different. And what happens is that we get our two distinct biological sexes. Okay. But we really came from, a generic blueprint. It's just, again, protein concentration. So human males have the XY chromosome and the human females has the XX chromosome. Now, the other 22, like I said, are like the autosomes. Okay. They're what we call autosomes. And because we have two sets, we call these a diploid organism. One from mom, one from dad. So the way that your textbook is going to write out diploid is by using this characteristics, 2n equals 46. The 2 representing the ploidy value, whether we're working with two sets or one. N is specifically telling us how many different karyotypes we have. And 46 is the total chromosome found in the nucleus. So in this case, 2n equals 46. We have 23 karyotypes from mom, 23 from dad. Okay, 23 chromosomes from mom, 23 from dad, and they're each their own individual karyotype. Okay. Now, when we are making our sperm and egg cells, we're cutting everything in half. So we call this a haploid cell because all genetic material is split in half. Okay. So here we can see the different sizes of the chromosome. You have the small ones, the medium ones, and the large ones. Those are 3 different karyotypes. So that means that when the gametes split, we should have 3 chromosomes at the end of meiosis. Okay. So fertilization is the union of the sperm and the egg, and we call this zygote. And we'll talk more about the term zygote in the next chapter. Okay. So meiosis. There are 2 distinct stages of meiosis. We have meiosis 1 and meiosis 2. Now take a pencil out. I want you to write this down. I'm going to tell you the main goal of each of these stages. For meiosis 1, you guys ready? For meiosis 1, the main goal is to take a diploid cell and turn it into a haploid cell. So we're taking total chromosomes and we're going to cut it in half equally. So you start with 10 chromosomes at the beginning, you should end up with 5 chromosomes at the end. If you started with 8 chromosomes, you should end up with 4 total chromosomes. Okay, that's it. That's the purpose of meiosis 1. We are taking a diploid and turning it into a haploid. In meiosis 2, the steps of meiosis 2 is identical to the steps of mitosis. And what is the purpose of mitosis? Is to take X chromosomes, X-shaped chromosomes, duplicated chromosomes, and splitting the sister chromatids into individual chromosomes. Purpose of meiosis 2 and mitosis is to take sister chromatids and split them into individual chromosomes. That's it, right? That's what mitosis is. We took X, we lined up on the metaphase plate, and then we split them. Now they're individual chromosomes. Okay, that's it. And all my students, when we first learn this, will always kind of forget, right? They'll think mitosis is like meiosis 1, and they'll just do that. It happens all too often, and I can't really say all my students because there are some that gets it right off the bat where they've had genetics before, but it is a very, very common error that a lot of students do. So I'm hoping that this kind of helps you guys. So if we take a look, we can see that when we go through the G1, this is currently in G1 phase, and we go into S and G2 phase, we're going to end up with duplicated chromosome. Now, in this example, this singular cell has 2 total chromosomes, right? But 2n equals 2, right? It's a diploid, one from mom, one from dad, so it's diploid. And the number of karyotypes we have is 1, but we have 2 total chromosomes. And notice what happens. We started with 2, and then we ended up with 1 chromosome in each cell. Now, why is it 1? Remember, when they're in their X shape, we still consider them as a singular chromosome because they haven't split yet. They're 2 chromatids, right? But 1 chromosome. It's only when we go into meiosis 2 and we split the chromatids apart, then they're both their own individual chromosome. So in this piece, we have 1 chromosome, 0 chromatids. Okay. So when you guys are drawing these stages out, draw it out and ask yourself in each individual stage, how many chromosomes do you see? Again, it's just how many shapes you see there, X or S. And then chromatids, you only ever count it, write this down, you only ever count chromatids when they're in the X shape. Okay. So it's going to go through the same stages. Notice prometaphase is not here. This is why I don't like it when they include prometaphase in mitosis, because in meiosis, they're not including prometaphase. So just know prometaphase is still here. I don't get why the book does this. It's very inconsistent. But anyways, you guys can read through this, but here's the key difference, and I'm just going to visually show you guys. Okay. What's the goal for meiosis one? Is to separate the homologous chromosome and make haploid cells. So we take a look. The distinct difference in meiosis one during prophase versus mitosis and meiosis two is that we are pairing up the homologs. We're going to pair them up, right? And during this pairing, certain things can happen where they can exchange their genetic material from one to the other. So notice now I have my yellow chromosome with a little bit of a red and my red chromosome with a little bit of yellow. Okay. This is what we call crossing over. Now, why does this occur? Again, variation. We need to have variations at the end of it. Now, once the homologous pair is up, nucleus breaks down as normal, centrosome still formed, does everything the same. What's the next? Well, they're stuck together right now. And what's going to happen is that they're all going to line up at the metaphyseal plate, same as metaphase. But instead of lining up in single file line where all the chromosomes line up in a straight line, we line up the pairs together. So notice we have 6 chromosomes. They're not all 6 lined up in a single file. Its homologs are all paired up. And then we split the homologs during anaphase 1. So now, when you look at telophase 1, okay, you can see we started with 6. We lined up 3 and 3, 3 on top, 3 on bottom, right? Because they're homologs, they're paired up. We split those homologs in half, right? So 3 goes to the bottom, 3 goes to the top. And notice we ended up with 3 chromosomes versus the 6 chromosomes we started with. So again, follow along, count it. 1, 2, 3, 4, 5, 6. Nucleus breaks. So 6 chromosomes, 12 chromatids. In metaphase 1, 6 chromosomes, 12 chromatids. In anaphase 1, 6 chromosomes, 12 chromatids. And in telophase 1, during or in each of the daughter cells, 3 chromosomes, 6 chromatids. We've halved it. This is where haploid cell comes from. That's meiosis. That's it. We are done. Okay. No, that's not it. But that's essentially it. Because if you go to meiosis 2, you're going to start with one of these daughter cells. Okay. And then you're going to run through the same stages. All of it's the exact same as mitosis. So notice here's one of the daughter cells. Prophase 2, nucleus breaks, chromosomes are still there, spindle fibers are forming, yada, yada, yada. Metaphase, we're going to line those chromosomes right in the middle, not paired up, just all in the middle, split them up, and then we have 3 chromosomes that we ended with in each of the daughter cells versus the 3 that we began with. But notice each of these daughter cells has different genetic material. Now note, here's a very quick rendition. I have prophase 1, right? I did crossing over. I split. That's the end of telophase 1 once we split. Now, one of these daughter cells right here, I'm going to take it. This is prophase 2. It's going to go through, line up with the equatorial, and then split. Now we have each of these in the daughter cell. But notice in this one, and I know it's very blurry because my image here, let me see. This one right here, okay, is yellow, yellow. And this one is yellow, red. They are not identical. These are different. Okay? So, watch an animation. Okay? So the 3 events that causes, or the 3 events that is unique to meiosis 1 is crossing over, homologous pair lining up, and the splitting of the homologous chromosomes. That's different from mitosis and meiosis 2. That's the 3 unique stages that are different. And then here, again, you guys can see what happens. For one cell, it splits into two cells. For one cell, it splits into four daughter cells that are not identical. These two daughter cells are identical. Okay. Now, last but not least, why do we need mitosis? For embryo development, we grow the cell, right? Again, we only need to use meiosis if we're building sperm or egg cells. But for all other cells, we're going to undergo mitosis. Healing, right? If you cut yourself, you got to build those cells back. Healing. Growing, if you're working out, tearing your muscle cells and you want to build muscle, mitosis, right? The reason why you have higher metabolic rate when you work out is because you're undergoing cellular respiration and it's heating up your body, and hence why you have a higher body temperature at night. Afterburn effect, that's what they call it. So all of this, I'm hoping is connecting. Okay. Mutations. Yes, you're going to have mutations in your DNA. We'll talk more about that when we talk about genetics. Okay. Three mechanisms that adds to variation. Independent assortment. Randomly assorting the chromosomes on the metaphysical plate. How you do it, who cares? As long as it's assorted properly, right? Single file or a homologs, doesn't matter. Crossing over is again, when their homologous pairs up and exchange information on one of the legs, and random fertilization. Whatever sperm hits the egg, yeah, that's variation. Okay. So to calculate the different variations we have, whatever number of karyotypes we have, we take 2 to that number. So if we want to look for humans, well, how many different variations we have? We have 2 to the 23rd power, which is over 8 million. Okay. Now, recombinant chromosome is just another name where you're recombining chromosomes, like different genetic material into one chromosome again. So this is what we call recombinant, is recombining the DNA from mom and dad into one chromosome. Okay. So each of these right here is a recombinant chromosome right here. So if you guys take a look at where the crossing over happens, that's a recombinant chromosome. Then you guys can watch this. Random fertilization, crossing over adds more variation again, and we're done. That's it. Hope you guys enjoyed this video. 1 hour. I think this was exactly 1 hour. We did it in 15 minutes. I told you guys. Chapter 13 is very short. So good luck. I didn't do any drawing. Hopefully you guys get that in my lecture. And hopefully if you guys already had that in lecture, review over it. Okay? Take care. Good luck.